Today, we're taking a look at a 2024 paper from Landwehr and colleagues, published in Nature Communications. This work introduces a platform designed for accelerated enzyme engineering. The research really gets at a central challenge in the field. How do you rapidly generate and, more importantly, learn from large sequence function datasets to enable predictive protein design?
So here's how we'll break down our analysis. First, we'll touch on the existing bottlenecks in enzyme engineering. Then, we'll introduce the study's cell-free design, build, test, learn platform. We'll see how it was used to map the enzyme McbA, guide mutant prediction with machine learning, and then experimentally validate those predictions before we wrap up with the broader implications for biocatalysis.
All right, let's start with Section 1. We'll be looking at the fundamental constraints that have traditionally limited both the pace and the scope of enzyme engineering.
The core problem that this research is trying to solve really comes down to two things. First, the theoretical protein sequence space is just hyper-astronomical. And second, our practical ability to screen variants experimentally remains very, very limited. So the central question the paper aims to resolve is the one you see here.
You know, traditional directed evolution is a powerful tool, but it has some inherent limitations. Because of low screening throughput, you can only ever sample a tiny fraction of the possible sequence space. This leads to a search that's path-dependent, meaning it can easily get stuck on a local fitness peak and might completely miss the global maximum. And crucially, this stepwise process often fails to identify beneficial epistatic interactions. That's where multiple mutations work together to produce a synergistic effect that's much greater than the sum of their individual parts.
Moving on to Section 2. To get around these challenges, the researchers developed an integrated platform that combines cell-free protein synthesis, high-throughput screening, and machine learning.
The entire platform is structured around the design, build, test, learn workflow, or DBTL for short. You can think of this as an iterative engineering cycle, where the data you get from each round of testing is used to systematically learn and then inform the next round of designs.
So let's walk through the 4 stages. First is the design phase, where specific residues are selected for mutagenesis. Second is build, where the libraries of mutants with defined sequences are generated. Third, you test these variants by screening them for activity. And finally, the learn phase, where all that resulting fitness data is fed into machine learning models that help guide the next design cycle.
Now, a key innovation here is in that build phase. By using a rapid, cell-free protein synthesis system, the platform completely bypasses the much slower cell-based methods for cloning and expression. This is what allows them to generate thousands of sequence-defined protein variants in less than 24 hours. The technical workflow for this is pretty streamlined. First, PCR is used to introduce the mutation you want. Then, the original methylated plasmid template is digested away, using an enzyme called DPNI. Next, the mutated DNA is circularized through Gibson assembly, and then amplified to create what are called linear expression templates. And finally, these templates are added directly into a cell-free expression system to produce the mutant protein, completely avoiding the need for cellular transformation and cultivation.
Okay, Section 3. Now we'll look at how this platform was applied to a specific enzyme, which is an amide synthetase called McbA.
The test subject they chose was McbA, an ATP-dependent amide synthetase. The reason they selected it is because it's known to be versatile. It has a relaxed substrate scope, which makes it a really good generalist enzyme to use, as a starting point for engineering into multiple distinct specialists.
So, the first experimental phase was all about establishing a functional baseline. To do this, they evaluated the substrate promiscuity of the wild-type McbA across an impressive 1,100 unique amidation reactions.
This extensive screening revealed some very clear substrate preferences. As the table shows, the wild-type McbA readily accepts aryl, benzoic, and cinnamic acids, but it doesn't do well with aliphatic acids. For the amine substrate, it was basically the reverse. It accepted aliphatic amines well, but had low activity with aryl amines.
From this initial screen, they found that the wild-type enzyme was already capable of synthesizing 11 different pharmaceutical compounds. The conversion rates were varied, ranging from just trace amounts all the way up to about 12%. This gave them a solid quantitative baseline for the engineering efforts that would follow.
All right, Section 4. With that baseline established, the next phase was to generate a data set large enough to train predictive machine learning models.
To create this dataset, they ran what they called a hotspot screen. This involved doing site saturation mutagenesis at 64 different residues located within the active site and its substrate access tunnels.
Then, the activity of the 1,216 single mutants they created was evaluated for the synthesis of 3 specific target pharmaceuticals.
The model they selected for this was augmented ridge regression. Let's break that down. It's a linear regression model, but its feature set is enhanced or augmented with the outputs from something called a zero-shot fitness predictor. The term zero-shot here is key. It signifies the ability to infer a variant's fitness based on large-scale evolutionary and structural data without needing prior experimental data for that specific variant. It's a way of incorporating generalized, external knowledge into the model.
This slide offers a good comparison between this ML-guided approach and the more traditional iterative saturation mutagenesis, or ISM. While ISM is a sequential, path-dependent search that can get trapped in local optima, the ML model learns from a broad set of single mutants, allowing it to extrapolate into unseen sequence space. This means it can screen huge combinatorial libraries in silico, which helps it identify beneficial higher-order combinations and significantly cuts down on the experimental workload.
Okay, Section 5. We've got the predictions from the model, but the question is, did they actually work? Let's turn to the experimental validation.
To establish a solid benchmark, a conventional directed evolution campaign using ISM was run in parallel for the synthesis of the pharmaceutical McbA. That campaign successfully identified a quadruple mutant that improved the conversion from the wild type's 12% all the way up to 96%.
And this chart lays out the results quite clearly. On the left, you have wild-type McbA at 12% conversion. The mutant found through the traditional ISM campaign reached 96%. And the key result is that the top mutants predicted by the machine learning model also achieved this 96% conversion level, but they did so with a fraction of the experimental effort.
When they performed a kinetic characterization of the best-engineered mutant from McbA, they found a 42-fold increase in catalytic efficiency, which is defined as Kcat over Km, when compared back to the wild-type enzyme.
The ML-guided approach also led to several key discoveries that really highlight its advantages. For instance, in synthesizing metoclopramide, the model predicted a highly beneficial mutation that the parallel ISM campaign had actually discarded in an earlier round. This suggests that the traditional ISM method had followed a suboptimal evolutionary path. Even more interesting, for synchicane, the model's best variant contained a mutation that was actually detrimental when it was a single mutant. This is a powerful demonstration of the model's ability to identify those beneficial, non-additive, epistatic interactions we talked about earlier.
To assess the platform's robustness, the workflow was applied to 9 different pharmaceutical targets. As you can see from this summary table, the results show significant improvements across the board. Everything from a 34-fold increase in yield for troxapid to a 30-fold activity increase for metoclopramide. This really proves the general applicability of the approach.
Now, one of the most critical findings of this study came from analyzing the results across all 9 campaigns. In every single case, the best quadruple mutant predicted by the ML model showed higher activity than a variant that was constructed by simply combining the 4 best single mutations. This confirms that the model is identifying true synergistic effects, not just simple additive improvements.
And that brings us to our final section, number 6. Let's zoom out and consider the broader implications of this integrated framework for the future of biocatalysts.
To sum it up, the advantages of this framework are significant. It's faster and more efficient, expediting engineering timelines and getting around the path dependency of older methods. It's scalable because it leverages low-cost, cell-free systems. It's also accessible since the machine learning models don't require specialized computing hardware. And ultimately, it provides a powerful platform for divergent evolution. That is, rapidly engineering one generalist enzyme into an entire panel of specialized biocatalysts.
The paper's central thesis is really encapsulated in this quote from the abstract. They state that this framework can fundamentally accelerate enzyme engineering by allowing scientists to iteratively explore sequence space and build multiple specialized catalysts in parallel.
This research provides a new paradigm for enzyme engineering, one that combines rapid, multiplexed data generation with predictive modeling. It leaves the field with a provocative and very important question. To what extent can frameworks like this one accelerate the discovery of biocatalysts for chemical syntheses that have, up until now, been considered intractable?